Hi, my name is Eric Crandall. Thanks for clicking on my lightning talk. Genetic diversity is the most fundamental level of biodiversity. As this session is about building genomics-informed conservation, I'd like to point out that the INSDC Sequence Read Archive, or SRA, is potentially a treasure trove of data for this purpose, containing 600 terabytes or 1.63 quadrillion base pairs of data from over 16,000 species. And it's growing exponentially, increasing its size by 50% in 2019 alone. However, the vast majority of these data are lacking the basic metadata that describe where and when a tissue sample was taken, making them useless for monitoring genetic diversity. In fact, we found that 86% of SRA datasets are lacking coordinates and collection date. This is taken from an audit of the SRA that was done by Dr. Rachel Toshilowski in 2020. Hey, Rachel. In the summer of 2020, with travel funds that couldn't be spent due to the pandemic, I recruited 14 students who had lost income or research opportunity and paid them to find some of these missing metadata. We are still analyzing the data, but here you can see the results. 863 out of 1,664 datasets were passed filters for human pathogens, model organisms, and domesticated species. Out of these, 504 ultimately were determined to be relevant to biodiversity, and not from lab cultures or breeding experiments. The black line in each bar graph shows the metadata that were already present in each category, so everything to the right of the black graph shows what we were able to add. Even after scouring publications, we were only able to find lat longs for 63% of BioProjects and collection date for 40%. Even after this intensive search, we only improved the percent of publications with both categories from 14 to 33%. All of this took a total of 2,000 human hours. We are in the midst of QCing these data, but we will soon add them to Geome, which is a metadata broker that connects biodiversity observations at the species and genetic levels. These metadata can eventually be crosswalked directly into the SRA in Mixus format. For more information, please check out our poster in the PNAS publication just out in August. Thank you.